Today I'm going to talk about scientific and regulatory considerations on in vitro release testing and in vitro in vivo correlation for complex long-acting drug suspensions. I would like to point out as a disclaimer, the content of the slides of the presentation and the subsequent discussion, those are all solely my personal scientific view and should not be construed as FDA policy or view. To begin, here's the outline of my talk. I would try to spend some time to provide an overview of IVRT methods and IVIVC about why, how, and what we can do. And when I was developing the talk, I was really hoping I could bring some, I'm not sharing. Can you see my slides?
Yeah, and this is my screen. Can you try again, please?
Oh, sorry. Okay,
That's
gotcha.
okay.
I guess I need to restart when I join the webinar. Okay.
There you go.
Okay, good. This presentation is going to be good because we already had all the troubles. Thank you. I was hoping I could give more direct answer to how we can get an IVIVC that is really useful for regulatory purpose. But then that made me realize where we stand and cherish this opportunity more to hear also from you in terms of what you think FDA can do better to support this effort. So I like to say we recognize the importance and the difficulties. So we are also doing research, trying to better understand what are the hurdles that prevent a successful development of IVIVC, and I'm using injectable drug suspension as a case study to try to shine some light on why IVIVC is challenging. So first, IVIVC starts with in vitro dissolution testing. So why do we do IVRT? One, IVRT has been an important part for supporting drug product development, such as use for formulation screening or to at least have some basic understanding of how a complex dosage form may release the drug. Then secondly, IVRT has been the gold standard for quality control for complex or conventional products to ensure batch-to-batch consistency. And then when it comes to generic or post-approval, for bioequivalence, we recognize for complex dosage forms, the conventional clinical PK or clinical endpoint studies can be challenging. So we are working on exploring alternative novel approaches. And IVRT has been a pivotal part as the in vitro performance testing. Sorry, just excuse me. Yep, then lastly, when one develops a successful IVIVC, then that can be used in lieu of in vivo tests to support a post-approval supplements, for instance, changes. Then next, how can we develop an IVRT method? What are the considerations or challenges? So first, we need to look at what is the product we're trying to develop a method for. What's the physical form? If it's a tablet, capsule, powder, semi-solids, right, transdermal patch, all those would dictate the suitability of a system. Then for dispersing system like injectable suspensions, liposome, emulsion, micelle, those things, there is a need to separate the released drug from the formulation and make sure we only are sampling the released drug instead of the actual formulation, which adds another layer of complexity. And then also when one develops a method, right, we need to consider if there is any physiological constraint and what is the route of the product. If it's ocular, we may have to consider the in vivo residence time to be more biorelevant. And if it's intravaginal, intrauterine, then those are generally very long-acting product and whether, we need to consider the physiological fluid composition of those things. Then lastly, there are also considerations to think about analytical related constraint. So for some drugs, if they are designed to have a rapid dissolution, then what is the most appropriate or feasible way to capture that? Or on the other hand, if it's designed to have a very slow and long-term release, then whether we should consider to have a method that could accommodate, to be able to have a low release rate to be more relevant to in vivo, but then balance the time course for the IVRT as well as the concentration at certain sampling time point. So all those things need to be addressed before we really could have a reasonable method. So this slide here just gives some examples of various dispersed, complex dosage forms. Today I'm mainly focused on the suspension, but then each system could have its own challenging. Like for abraxane, for instance, is a protein small molecule complex. The drug is actually designed to have a rapid dissolution in vivo post-administration. But abraxane itself has extremely low water solubility, and how you balance that rapid dissolution design versus the low water solubility, this is one challenge we are facing and we're still working on. Then for suspension, there are other things I'll further elaborate in my later slides. So in terms of methods, setups, this slide, I try to give a comprehensive summary or list of the methods we have seen are the relevant adapters. So FDA, we don't have a hard line in terms of you have to use USP apparatuses. But in general, if you go through our guidances, we generally would say we recommend whenever feasible, you consider USP apparatus because they are more standardized. But when it comes to complex dosage forms, it's very hard to find one size fits all scenario. So for complex suspensions, what we have seen the most are USP apparatus 2 and USP apparatus 4. So each of them have their own pros and cons. And related adapters, we have seen, for instance, the dialysis stack. Then the pulsatile microdialysis I'm showing here, that was a system which was designed actually more to capture, for instance, the ophthalmic rapid ophthalmic product to capture the initial release in a short time period, because the system gives a significantly increased surface area to volume ratio, allow the initial capture of the dissolution. And then the lower corner, which I have, yeah, and then this adaptive perfusion system, I don't know if any of you have actually read a paper published by FDA lab scientists. The column used here, the TFF system, is conventionally used for peptide protein purification. But this system was developed a few years ago to conduct a dissolution for an emulsion system, which is very useful to really be able to give enough driving force to drive the drug release from the oil droplets. And then for suspension, there are other efforts going on in advancing the IVRT setups. Then we know there are miniaturized flow-through cells. Then the Caesar system by Pion is something developed for subcutaneous injectable suspension that you could have a potentially more biorelevant media, and also you can monitor the image of the suspension. So we generally, when we do research, we're trying to explore all kinds of methods to know what's out there up to date. And we're open actually to really see efforts and explore new setups with companies. So when it comes to IVRT validation, which is one thing we commonly see in new drug and generic drugs applications as deficiencies, is we don't see enough or adequate validation. So what we are looking for, in terms of reproducibility, we're looking for precision and robustness. And then another critical part for the IVRT validation is to demonstrate discriminatory ability, to show the method is sensitive. So you could, develop a target formulation and also another set of target formulation with known changes, such as the quantity of drug loading. Then you could show the method is sensitive to those known changes, and you'll be able to capture them properly as you expect. Then for selectivity, you would, propose a list of critical quality attributes and justify why you think those are critical, and propose a range of changes that you think are relevant or reasonable, such as for injectable suspension, that one commonly evaluated CQA is the particle size. We do recognize sometimes it can be challenging, right? Depends on how much change you introduce, whether your method be able to pick it up or not. But then I think the bottom line is we want to see enough rationale and justification. Even you have to propose a bigger change to be able to demonstrate or validate your IVRT, then you need to give us enough justification why that range is reasonable, so we can go from there. What we often, what I have seen missing is company just give us the results, but they don't really tell us why and how they get there. Then in terms of generally speaking, those like reproducibility, discriminatory, those validation, those are essential. Those like things we have to know, must know. Then there are also things that's good to know. So what are good to knows in general? The release mechanism, if we could have some more data to really help us to better understand the product, how the drug may be released in vitro and in vivo, then IVIVC that comes to play, right? If you have explored any in vitro in vivo correlation during drug product development. Then for IVRT, conventionally buffer probably is, and plus some surfactant if needed, are the most commonly used dissolution media. But nowadays, since we are trying to promote IVIVC, then how to make the method more physiologically relevant is another topic or consideration. So the question then is, where can we start for developing IVRT method? If it's a brand new product, then it might start with in-house early development, or if this dosage form is not new, but the drug substance is new, then you could look into maybe literature or FDA dissolution database. Sometimes can be also helpful. So here I have a table just giving you a few typical examples of what you may see in the FDA dissolution database. So when it comes to the database, sometimes we get feedback saying, oh, you see, we go to the dissolution database, but there is no method listed. So what happens with this is in the FDA, there is a designated group. They periodically update the database whenever a method is deemed appropriate to be shared. Then also sometimes, like for instance here, in terms of just giving a hint, we consider this as a complex product, and likely you will need an IVRT method for QC purpose or for other purposes. Then this could give a heads up. You would keep that in mind. You probably will need an IVRT method that is part of your submission. But then for some products, as I'm showing here in column in rows 2 and 3, we give quite detailed information on a particular method or setup or even sampling time point. But those are by no means like you have to follow exact. This again is more like a guidance, a reference. Then lastly, like this case, triamcinolone acetonide, we don't really give anything very, very specific in terms of the experimental conditions, but we do provide a general recommendations for you may consider USP 4 or 2 if applicable, but then we are also open to any other methods. So the next few slides, I'll switch gear a little bit to talk about some general concepts or thinking about IVIVC. So for in vitro in vivo correlation, per FDA's general guidance, there is a guidance for industry, although it's for oral modified release products, but the fundamental principles actually are still applicable to non-oral complex dosage forms. So IVIVC is considered as a predictive mathematical model that could describe the relationship between an in vitro property of a dosage form and a relevant in vivo response. So in the guidance, there are different levels of IVIVC. Level A represents a point-to-point relationship between in vitro and in vivo profiles. And this is the only level that can be used to obtain biovivor, although I'll talk, also the guidance also emphasize actually on multiple level, multiple level C, which I'll talk in a little bit. Then level B is a statistical moment analysis. It's a mean in vitro dissolution time or with either a mean in vivo residence or dissolution time. This may be useful to support a dissolution in vitro IVRT specification. And then level C correlation establishes a single point relationship between the in vitro and in vivo. Then here comes the multiple level C that correlates multiple dissolution time points to one or more PK parameters. So generally, the thinking is if one be able to achieve multiple level C, then there is a good chance they actually could possibly achieve level A. So it's highly encouraged if one could achieve multiple levels C, then they should further explore to see if they be able to get a level A. Then for level D, or the guidance don't really call it level D, is it says rank order. That is useful for supporting early formulation screening during development, but at the moment is considered not useful for regulatory purposes because it's only a rank order correlation. So why do we want to develop an IVIVC since it's actually a challenging task? So from product development standpoint, an IVIVC can guide formulation development, screening, as well as actually process development changes in various stage or manufacturing in process control, for instance, once you really understand your dosage form. And it could help and support validate the use of an IVRT method and help set clinically relevant dissolution specifications, which I think I'm not from biopharmaceutics, so I don't want to speak on their behalf, but this is something generally is like preferred or recommended. Then when a level A IVIVC is established and validated, then the IVRT as a standalone study can be used as a surrogate for bioequivalent studies when a pre-approval or post-approval changes occur. So common issues for IVIVC, I try to look into if we, have a good summary of the common deficiencies of IVIVC. And up till today, we still really just have very limited number of cases that company have submitted data on IVIVC, and majority of them are still limited to oral dosage forms. For injectable suspension, I think there is a few. There is one successful in human IVIVC for injectable suspension, and there are also a couple preclinical IVIVC that was submitted in the NDA submission. So what we have seen in terms of common issues of failure, one is company sometimes give us an incomplete IVIVC report. which doesn't allow us to really do proper assessment. So like I said, even for IVRT report, validation report alone, very, very often, at least for the generic side, we don't see the adequate validation report to allow proper assessment. Then use of formulations with only one release rate for the proposed IVIVC without evidence to show the dissolution is condition independent. I think for complex injectable suspension is probably very rare or challenging. One can develop an IVIVC rely on only one release rate. Generally, the number of formulations you evaluate for your IVIVC also could have an impact on later how useful the IVIVC can be. So generally, the successful cases I have seen for suspension, at least the 3 or more different formulations with varied particle size were investigated. Then also for this is more probably for generic IVIVC. When they try to develop the IVIVC, they would put the reference product as part of the initial group of the formulation, which is not appropriate because for the group of IVIVC, you are exploring the formulations. You should know what you are testing, right? Those are the things, the formulations should be developed by you, not from a different manufacturer. Then the last commonly seen issue is companies do not really adequately assess the external or internal predictability. And like the IVIVC model did not really accurately predict the plasma concentration or the PK profiles in the range of selected release rate. So IVIVC is challenging for in general and for injectable suspensions. It could be more challenging compared to conventional like oral dosage forms. So one thing we're trying to go back to the fundamental is really to understand what are the things, right, that could impact the drug release or from the injectable suspension. So what we learned, again, this slide by no means is exclusive points, but a few key aspects we have observed that drug in vitro release and in vivo absorption PK can be principally affected. One is drug itself, because the drug substance suspension is just the drug substance suspension, right? There is no really controlling inactive. So the drug dissolution kinetics or itself, its property is the key here. So the drug, its own solubility is one critical factor, and then the particle size, and as well as I have a case study to show the agglomeration state that could have a huge impact on how the drug may be released. And then also the surface and morphology features of the drug crystals, again, that could be giving a downstream impact, right, on the particle size and agglomeration state, as well as other formulation variables, such as how much drug you put into 1 ml, how many drug particles, how concentrated it is, and also the viscosity of your dispersing media system. And then another thing we start to realize the criticalness or the importance is the local tissue response post-injection, the local inflammatory response at the injection site. Post-injection, one would well experience acute and chronic inflammation, and the acute edema and swelling at the injection site could result in a rapid increase in the drug level in plasma following administration, although that depends on the drug solubility and the particle size post-administration. And then a subsequent chronic inflammation reaction could do the opposite effect, like retard the drug dissolution and absorption rates, results in a flip-flop PK absorption profile. So I have seen literature did some like initial preliminary investigation, and there are evidence showing, for instance, when the particles are big enough, they could result more severe chronic inflammation reaction. Then the fibrosis encapsulation layer formed around the depot or the injection site could really significantly retard the drug release or permeation into the tissue to be absorbed. So next a few slides, I'm going to dive into my case study and to highlight why IVIVC for injectable suspension is challenging. I'm using triamcinolone injectable suspension as the model product. So we recognize this. So the picture here showing this is the triamcinolone acetonide product when it's on the shelf. So you can see the formulation is segmented, and this part represents the segmentation final volume of the suspension. And this B picture here is after redispersion. So you can see the segmentation volume is quite significant. And depending on the measurement condition, the particle size could vary from 2 to 10s of 2 micron to 10s of micron. And this particular formulation is designed to stay at the secondary minimal flocculation stage to give enough physical stability to allow redispersion post the shelf life. So it forms flocculates, and then this sedimentation is loosely packed flocculates with different particle size. And one thing, not just particularly for this product, I would think is a general observation for injectable suspension is a highly variable PK. It's for this particular one, as I'm showing here, for product A and B, they have a bimodal PK. You have an initial increase, which is the Cmax post administration, but then you also have a little secondary shoulder after the initial decay, then you have a secondary. So all those things complicates the IVRT method development and how you would handle, because in general, this in vitro dissolution is more like a continuous diffusion kinetics. So here, this slide shows you the optical image of this formulation, and you could see there are like primary, we'll call that primary particles that are very fine small particles, while you also have the secondary particles as agglomerates. So the particle flocculation is a common phenomenon in suspension product. Although we know for newer generations, this is a key formulation consideration and a choice for a company to decide if they actually purposely make the formulation as a flocculated system, or they could use other combination of stabilizer to avoid it. So what we realized what's the impact, right? So what we realized is the particle flocculates. Then when it undergoes shear during the IM administration, for instance, the injection rate and needle gauge actually could affect what's the particle size when they enter the body. So what we are testing the method here is actually a novel method for the system, and we are trying to mimic high shear and low shear for administration to know when by doing this we would introduce suspensions in different agglomerate state, therefore different particle size, then to see how that would subsequently affect the release. And then each system still we need to have some level of stirring just to make sure we can properly test and monitor the drug release, which I would say is not present as much in the body. And another thing we explored is the non-sync condition. Although conventionally, sync condition has been emphasized in many different cases when one develops an IVRT, but in this case, the non-sync condition is more relevant to in vivo, and our belief is if this is done in a sync condition, we might not see what we have seen. So move on to the next slide here. So in terms of the particle size distribution, when the formulations are introduced into the measurement by injection as they would be in a clinical setting. The particle size is represented by the red curve and is smaller than the low shear, which is introduced by pipetting. Then in terms of dissolution, you can see the two groups actually have very distinct release kinetics, as shown on the right hand, in terms of percentage release. So this method was able to actually properly capture and separate the two groups of difference. So the shear rate would affect how the particles, the particle size when they are first entering the system. And then subsequently, they affect the dissolution rate. And if this is in vivo, then it will further subsequently potentially affect the local drug dissolution and bioavailability. So the deflocculated primary particles can exhibit more than 4 times higher the dissolution rate than the flocculated system. So this here picture just gives you a snapshot of how the system is set up and what it looks like. So because the dissolution is actually fairly quick, right? If you look at the previous slide, it's in minutes. So conventional sample and separation can really keep up with this sort of dissolution setup. So what the lab staff they did is they basically assembled particles, the master sizer for continuous inline monitoring for particle size change. And then they also introduced a UV-Vis system from Pion to in situ continuously monitor the drug concentration during dissolution. So this is a novel setup. And this is something I found interesting is we have been publishing papers on very novel IVRT setups. I think companies do appreciate the effort, but at the same time, sometimes I think we introduce a little nervousness, like people feel like, what if I cannot do it? So again, I think what we are trying to achieve here is to demonstrate some feasibility of the novel method and also send the message that we are open to seeing novel methods, as long as we get enough information to understand, we are open to have more discussion at this front. So what I like to show you in this slide here is quite interesting, which I don't think other paper have discussed, is when you have a low shear rate, then you introduce the particles as flocculates, right? So they begin with bigger particle size, and then through the continuous particle size monitoring along with the in vitro dissolution, what you see is you see a decrease in particle size. So the flocculated suspension, they exhibit large shift in particle size distribution during dissolution, meaning they actually forms a little cluster and then they sort of shrink, right? They shrink from outside in to dissolve the drug and release the drug. Well, on the other hand, when they are introduced by needles and they are already at their primary particle stage, the dissolution is much faster, but then also interestingly, they only exhibit minimum change to the particle size distribution during dissolution. What we see goes down with time is the counts of particles instead of the actual particle size. So that's why those primary particles, they provide much more dissolution. They just sort of disappear on individual. But then the particle size as a general, as an average, or you don't really see an obvious shift, which is very interesting. In terms of the release mechanism, although fundamental mechanism is still drug diffusion, right? But then in terms of how they actually would dissolve from which population and the method, that gives an interesting aspect, also considering in vivo. So what we are seeing here compared to what's happening in vivo, our belief is that it's still significantly underestimate the difference when you have a difference in injection rate, for instance, for different subjects, or if you use a different needle gauge. For newer generation, the needles and everything, those at least are defined, but for some other older generation is not, although clinical setting, it may still well established. But this is just some interesting factors to sort of keep in mind. And when we explore IVIVC, and to better understand what's happening in vivo post the administration and what's the optimal formulation design, particle size for injectable suspension, this can be an interesting setup to better understand the system. And this is the last slide about the work we have done on the IVRT for suspension, and we teased out the initial 15 minutes just to highlight the initial dissolution rate, the flocculated versus deflocculated. It is significant difference. Just simply by changing the shear rate, how you inject the product into the system. So to summarize my talk, we think selection of an IVRT method depends on physical form of the product, and complexity in IVRT generally is due to the need you have to separate, right, release the drug from other constraints. And in order to design a good IVRT method, we really need to have a good understanding of the formulation and how the formulation is manufactured. And formulation with intentional variability, variations are good testing samples to verify if your IVRT method is discriminatory and reproducible. And understanding the mechanism of release and factors that controls the release in vitro and in vivo can guide the development and improvement of IVRT method and IVIVC. And lastly, I'd like to see FDA is working on opening multiple different channels to have early engagement with industry to facilitate development. So if you are looking at something novel and you have done enough work, is always highly encouraged and appreciated from our end, to really have an early engagement to learn what you are doing and see what we can do to further support the effort. So with that, I would like to thank my FDA team, my external collaborators, and the funding source. I'm privileged to present the work we have done, the improved understanding we have received, but the work I presented on triamcinolone and it was carried out by Dr. William Smith and his supervisor, Dr. Xiaoming Xu. So they are the ones really did all the work and actually helped me with some of the slides. And also my colleagues in my division, my supervisor, Dr. Kozek, my team members, for the constant discussion, and we just enjoying really the work and also the interactions with industry. And throughout the years, also I'm privileged to have many wonderful external collaborators. Then in this particular area, I have collaborated with Dr. Van Burgess, Dr. Chen Bao, who is here today, and like to thank GDUFA really for, at least for generic, GDUFA really made a difference in terms of relief, research, and also we have got internal FDA grants to support our research. So with that, I will be happy to take any questions you may have. Thank you.
Thank you so much, Ian, for your wonderful presentation. It's very informative. So please submit your question to the Q&A box. And yeah, we may start from some kind of question. And so first, I want to introduce our panel list. So the chair of this IQ Consortium working group, Stephanie Barrett from Merck, and our co-chair, Venkat Koganti from the BMS, and our co-moderator, Pratik Shah from GSK. So let me start with the first question because the time is very limited. I just want to do as fast as possible. So the first question is about the IVRT method development for the LAI suspensions. So what is the role and expectation of the FDA dissolution database?
Sure, thank you for that question. So FDA database is generally served as a good starting point if you think the method is useful, but by no means is a mandatory binding recommendation. So we always encourage companies to come up with their own method as long as they provide enough justification and validation. Another like maybe misunderstanding we're seeing is if they find the FDA method is actually useful or suitable for their own development, they would just directly use it without any validation, which is another common deficiencies we see. Even when you think FDA method is suitable for your system, you still need to go through the validation using your own in-house very target formulations to demonstrate the suitability.
Thank you. Pratik, please. Pratik.
Yes, hi, Dr. Wang. Thank you very much for a very good presentation. And maybe a couple of questions based on what I heard. Just give me a second. Yeah, so I think the dissolution database that you showed, right, the AFDA's, it's more probably QC methods, right? And not necessarily biorelevant, the examples that you showed, and those were what, sink conditions?
Yeah, I mean, so again, FDA method is can be used to fit your purpose. I would not say it's only feasible, like suitable for QC, but most of the time for majority of the applications, right? IVRT is used for QC purpose. But then if you want to explore IVIVC, then like I presented here for the suspension case, we are more open to non-conventional, non-sink condition. Like in this case, if you use the sink condition, you may not be able to actually distinguish the two, which in reality, we do believe the different flocculation stage of the suspension contributes. Maybe whether it's dominant or not, that needs further, investigation. But at least we believe that contributes to the observed PK variability.
And you need to almost show close to complete release. Specifically, if you're using this for IVIVC or to establish IVIVC. No?
Yeah, so I think that can be maybe a loaded question in terms of completeness, right? So for IVRT in general, if a dosage form like a suspension is believed is feasible to achieve a complete release, meaning more than 85%, then the expectation is you should. But then on the other hand, for instance, ophthalmic emulsions or ointments, those dosage forms, we know the dissolution to achieve complete conventional completeness is very challenging, right? So as long as company give us justification as for why the time course or the percentage they have achieved should be considered as reasonable, then it can be, acceptable if the justification is adequate. But then back to your question about IVIVC, whether you need to see a complete release, I think that also depends on your PK profile, right? So for long-acting injectable suspension, just not considering IVIVC, just for clinical study alone itself, we actually get questions in terms of how long we need to sample, whether we can truncate the in vivo PK because the time is just so long. Then again, whether we could or not, I think the good part, at least personally, I'm hopeful because I'm working on it, is we are really trying hard to get more data and understanding as for why we could truncate, for instance, at a certain time point. But then the question we face internally from the assessment discipline is if you don't carry it out until the full elimination, what are you missing? What are the risks, right? So I think if we have enough understanding and justification as for post a certain time period, right? If the system fully settled and the dissolution is more just a gradual, dissolution from the depot, and you don't expect any sudden changes and also other, in vivo factors, that's something we, at least personally, my team, we are trying to work on more, like to better understand when and how we can truncate. And then that's, IVIVC.
No, thank you. Maybe just while I have maybe just one comment. I mean, obviously, with the emergence of long-acting injectables, maybe this is more of a wish list. I mean, if you think about it, right, we have IVIVC guidance, but that's for extended release oral dosage forms and so on and so forth. Really, for the industry folks who are in this space, maybe one of the wish lists eventually is, could we actually have some guidance, like the FDA has guidance documents around everything, around long-acting injectables and IVIVC specifically? Because as you said, it is a complicated topic. If you have an IVIVC, you also mentioned something about edema. It's probably saying edema is not playing a role or whatever. So I think eventually, I think it would be really useful if we can get to, from the agency, some sort of a guidance document around long-acting injectables specifically. And I'll stop there. Yeah, that's more of a wish list. Thank you.
Yes, thank you, Yan, the Pratik. And so we go to our next question. So what's the benefits and limitation of preclinical IVIVC for the drug submission period stage?
Yeah, I think preclinical or animal IVIVC is like a topic that could trigger many different opinions, right? Personally, I think is something very helpful, although in general, if you want to apply an IVIVC for any regulatory purposes, till today, our response to industry is you need to give us human data. But then on the other hand, what I see this is one could consider IVIVC animal may not be the pivotal or standalone. What I would approach if I'm in the industry, for instance. So one thing I want to make it clear is sometimes I feel like our guidances are great, right? Could give you a guidance, but at the same time, even internally, externally, I feel like the guidance also could become a box. People trying to stay in, and it's non-binding, but everyone is so nervous about stepping out of that box. But the reality really is, unless something is a regulation, which means it's a law, then everything else is negotiable. I know it's my personal, wishful thinking, but that's also true. At least how we are trying to handle and work with industry is come to us, be bold, and like negotiate. So for animal work, what I would approach, for instance, if today I have a major change, right? For suspension, for instance, then we know in principle, in vitro only approach may even be feasible with enough justification. So I would try to, for instance, propose an alternative in vitro approach, but I use the preclinical IVIVC as my part of my justification or evidence to show the design space for my formulation. Why I think the change will still stay within that design space. Well, therefore, it should not lead to any concerns in inequivalence in vivo in human. So that's how I would personally, I hope my FDA colleagues will feel like, are you crazy talking about like this? But that's what I believe personally, how, we could potentially utilize. And that's something we're also trying to push and work on, having more data and then also have more internal discussion.
Thanks, Yan. Yeah, that makes a lot of sense to me too. Okay, because time very limited, so we have a lot of questions. Thank you, the audience, for all of the very great questions. So the next question is for the QC release testing of LAI suspension. Is it advisable to follow FDA guidance on extended or modified release for setting specifications?
Yeah, I think so. For that, that's really out of my work scope. Is more like biopharmaceutics, but generally, I think the scientific principles stated in the guidance that should be applicable.
Okay. So next question is sensitive part of discriminatory. How is sensitive used to demonstrate the method being discriminatory? Thanks.
So the sensitivity part I mentioned, so the discriminatory, we could consider that as sensitivity and selectivity. So sensitivity, we more refer to if you have a change in the component of your formulation, then one obvious choice or easiest is to change the drug loading or the amount of drug, right, in concentration. Then whether your method be able to pick it up. Then the selectivity is more like we're trying to define, right? Then so the selectivity is more towards the manufacturing. If you have a manufacturing change that could potentially lead to a change in your formulation characteristics, even your formulation composition is still the same, then whether your method be able to selectively discriminate the formulation changes. So discriminatory, you could just put as a one word, not define it into sensitivity or selectivity, but the bottom line is you need to give us data to demonstrate your method can distinguish the target formulations from the ones that with unwanted changes or changes that will lead to difference in use.
Thanks. So next question is the lack of IVIVC in LAI stem from the fact that measuring the plasma concentrations of released drug from the formulation is extremely challenging?
So I'm not very clear in terms of measuring plasma concentration from released drug is challenging. I mean, for injectable suspension, I don't think that's a very challenging task to determine your drug plasma concentration. What is challenging when I think the dosage form is like the iron products or liposomal products. Those are IV administered, and to separate the drug from the formulation, that can be challenging. And that's something also I think my office will have worked on, and certain area will continue to work on. But for injectable suspension, I think the challenge here is, although it's supposed to be a continuous dissolution, like I presented here, you could have a multiphasic or biphasic in vivo PK, and how you potentially have an in vitro dissolution that could more closely mimic that particular kinetics. I think that's the challenging part. And also for suspension, how do you have a method or condition that could better represent the true particle size post-administration? That's another challenge.
Okay, the time is very limited. I think we can extend our meeting to 12:05, give another 5 minutes, and maybe take another last question. What is the expected in the method development report? Is this a must-have or nice to have in a first-in-human IND? What is expected in the method development report? So this, I'm not quite clear. So the method development report, do you mean that AD method development report? And is this a must-have or nice to have in the first-in-human IND? Yan, do you know the question? I'm sorry, I'm not quite.
Yeah, maybe it is referring to Yan for phase 1 IND.
Okay.
How much do you need in terms of the dissolution method development? I think that's what he's probably referring to. Must have versus nice to have with regards to a dissolution method development for these injectable suspensions, specifically more for phase 1 IND time. I think that's what it's, I'm just trying to, yeah.
Okay, now that question does make sense to me. But unfortunately, it's more like the OMB review discipline. So if there's a way I can get back to this question through written communication, I will do that. I don't want to misspeak for the review process.
Okay, yes, thank you so much for all of the questions and very nice answers. And now I will invite our chair for this seminar, and Stephanie Barrett from Merck, to give a wrap-up closing remark.
Okay, I just want to say thank you, Jan, for taking the time to put these slides together and sort of get your thoughts together. clearly there's a lot of interest when it comes to long-acting injectables. I mean, our particular IQ group has, I think, 13 different companies as part of it who are all thinking about long-acting injectables day and night. So we appreciate that you took the time to put these slides together and come talk to us, and of course, all of our 274 best friends who dialed into this webinar. So very much appreciated. I think it's going to be very helpful to get us all on the same page and sort of thinking about the same background documents and examples that you could provide here. And we very much appreciate it. I'm sure this is one of many interactions that you'll see for most of the faces that were on this call. Very, very much appreciate you taking the time to do this. And thank you to everyone who dialed in. There is an opportunity. So Yan did mention that she could respond to some of these questions later on. We can all continue this correspondence. So feel free to reach out to myself or Pratik or Kwenying or anybody that you recognize on this call, especially those that are part of this IQ group. We can definitely communicate that information afterwards if you still have questions or want to be part of the team. So thank you, everyone. Have a great weekend. For those who are almost ready to get there, thank you.
Thank you.
Thank you.
Thanks, everyone, on behalf of the
Thank
IQ Consortium. Have a great weekend.
You too. Thank you. Bye.